First of all, it has very little mass compared to the rest of the body, but it spends about 20% of all the energy that we have. So this per se is actually very interesting, and because energy is what drives basically any biological life, right? So how come such a tiny, small piece can actually drive so much energy, right? It's not only related to the number of cells that we have in the brain. So it's really, we have almost 100 billion cells, just the neurons. We have more than that if you count all the other cells that we have in the brain. And they are all connected to each other, so we have about 10 to the 15 connections, just counting the connections around the neurons, which are kind of the most typical cell that we have in the brain. It's not the only cell. But what really is interesting about the brain is that it has a very unique functional organization, and we can describe the anatomy, we can see the anatomy, we can actually take images of it, but it does not explain all the functions that we have in the brain. And that's typical, that's unique to the brain. You can actually look at the heart, you know how it works, both from the structural part, you know how it works from the anatomical part. You can look at any organ that we have in our system, we know we can explain that, but we cannot explain how the brain works still, right? So I feel like that's really fascinating. And over the last decades, we have developed actually very good ways to look at the brain when it's functioning. And we can do that with a lot of different techniques, right? Before, I would say 1980s, the best way that I could actually see how the brain works is open it. It's completely invasive. After 1980s, we start to develop new techniques that we don't need to open the brain and see how it works. So, and these are actually some of the techniques that we have. We have electroencephalography. It's probably the most known technique to study the brain or to see the brain at work. It's basically, that we call it EEG. So basically what it does, it just has a lot of electrodes in the brain, and as the brain works, the neurons just spikes, and that actually has, as a consequence, difference in potential, electric potential, so you can actually measure the difference in electric potential between different regions of the brain. And that's sort of related to the activity of the neurons. Most recently, we can actually use the radiation from positron emission tomography to look at different functions of the brain, because, well, you have a lot of radio tracers that can trace different molecules working in function. So you can actually have contrast based on radiation on that. The problem with PET is because it's not invasive in the sense that you open the brain, but it's still not the ideal technique because it needs radiation, right? And it's not appropriate for some reasons and for some populations. What really is kind of the top of the part is functional magnetic resonance imaging. So the idea of MRI is just look at the nucleons of the protons that we have, and based on an external magnetic field, you can actually you can actually see the brain. That's from the 1970s. But also, because hemoglobin, which is a molecule that you have in the blood, and it's totally related to function. I'll talk a little bit later on this. It has magnetic properties. And basically, oxyhemoglobin, the hemoglobin with oxygen, is diamagnetic. And the hemoglobin without the oxygen is it's not diamagnetic. So that induces a kind of small disturbance in the field that you can actually measure. So you can use oxygen from hemoglobin as a tracer to see when the brain is working and where it's working, how. Right. So these are actually famous techniques. What we actually developed in Campinas is a different type of field, also based on hemoglobin, which is basically, we call it diffuse optical spectroscopy. And the idea is basically like the regular optical spectroscopy when you actually have a laser light source that shines light, and you have a sample here. So if this sample is a homogeneous sample, you basically don't have scattering, it's just absorption. And that's the kind of chemistry 101 that we have in labs. So the amount of light that actually comes out is related to the amount of absorption you have in the sample. However, if your sample in the median has a lot of heterogeneity there, then you have a lot of scattering. And if this scattering is way higher than absorption, what you see, macroscopically speaking, is not just a standard propagation, but it's kind of like diffusing through the sample, right? So, and that's what the names diffuse optical spectroscopy comes out. And basically, because it's very heterogeneous, what you can do, you can model this. So basically the light fluence, which is proportional to the intensity, you can write a diffusion equation for this. And this diffusion equation will depend on both the absorption of the median and also the scattering that is related to a diffusion coefficient of the median, right? And by just measuring the amount of intensity that you detect and comparing to the model, you can fit that, and you can actually know the absolute values for absorption and scattering. If you know the absorption, you know the sample you have, you know what chromophores you have that absorb the light, what you can actually do is you have access to the concentration of the molecules there, right? So that's basically the same idea. That's basically the simple idea of optical spectroscopy. And why this is useful? Well, if your sample is tissue, biological tissue, and your light is near-infrared light, around 700 to 900 nanometers wavelength, what happens is actually the major chromophores that absorb light, it turns out it's hemoglobin, both hemoglobin with oxygen or oxyhemoglobin, or hemoglobin without oxygen, which is, we call it deoxyhemoglobin, right? So they absorb in the near-infrared region between 700 and 900. They are the major chromophores. If you go higher than 900, then water starts to absorb a lot, and we are basically 80% water, so no light will come out. All the light will actually just be absorbed by water. If you go less than 700 nanometers in light wavelength or light source, you have a lot of absorption, so no light gets backscattered again, okay? So this is kind of an optical window where you have scattering, a lot of scattering. You still have absorption, but it's smaller compared to scattering, right? And well, if you have a way to separate the contributions from oxyhemoglobin and deoxyhemoglobin, what you can actually do, you can measure absorption, and then you can separate them. You have an access to the concentration of oxyhemoglobin in tissue and deoxyhemoglobin in tissue, right? And the way any biological tissue works is, well, you have the cells in tissue, and they actually activate, so you have a lot of activity from the cells that demands energy, and energy in biological tissue is basically ATP, and what you need to actually break ATP to generate energy is glucose and oxygen, basically, right? So if we have glucose and oxygen, you can generate energy that, well, the cells demand to actually keep working. So oxygen comes from the hemoglobin, right? So by measuring the concentration of oxyhemoglobin that you have in tissue and the concentration of the fraction of deoxyhemoglobin that you have in tissue, you have basically access to really the energy demand in that region, right? And that's totally related to functional, how the tissue is functioning there, right? And that's actually very useful to study in the brain, because basically it's a very easy way to shine light on the scalp of any head. And light diffuses through tissue. Part of this light comes out, and if you detect that, you can measure absorption. From absorption, you can measure the concentration of oxy and deoxyhemoglobin, so you have an idea of how the brain functions in different regions, right? So that's basically the idea. You can monitor brain function based on hemoglobin concentrations, That's basically the same idea. It's the same idea of MRI. The difference is actually in MRI, the signal comes from the magnetic properties of hemoglobin. Here, the signal comes from the optical properties of hemoglobin, right? And well, so what you actually trace, neuron activity, I basically briefly just explained that, right? So if you have the tissue inside, you have neuron activity. If you have an activity there, you have metabolic demand, so that will induce a hemodynamic change, a change in blood, and therefore a change in oxy and deoxyhemoglobin, right? So this is kind of a very simple experiment that can convince anyone that you actually can measure brain function. It was based on rats about 10 years ago. So basically, if you look at the brain, different regions of the brain are related to different functions of the brain, right? And in the case of a rat, you have these whisker barrels, and these whisker barrels are related to smell for the brain, and it's related to one very specific part of the brain cortex of the rat, right? So if you actually look, and this is very well mapped, so you can actually look at the very specific region of the brain where this barrel is related to, and you can induce an activity by just stretching the barrel of the rat. So if you do that and you look at the right region of the brain, what you see is an electroactivity, right? It's basically the cells, the neurons, just spiking all the time. That's basically action potential. It's kind of a low frequency action potential, right? So it's related from a set of different action potentials of the brain. And if you look at the same region, you can actually monitor the concentration in oxyhemoglobin going up, because as the brain keeps working, you need energy to supply that, and then you need blood coming in that region. That will tell you that you have an increase in oxyhemoglobin in that region, right? And at the same time, you have a decrease in the oxyhemoglobin, you have an increase in blood flow. So basically that's the idea, and that's how we actually monitor optically or with MRI brain function. Just looking at hemoglobin concentrations going up and going down. Every time oxyhemoglobin is going up, we know that there's a need of oxygen in that region, so we know there's a neuronal activity in that region, right? And you can monitor different regions of the brain at the same time, right? And going really quick from a rat to human brain, you can actually do that to really understand how the brain works with function, at different ways to function. So you can go just for a very example, very simple, we call it primary tasks. So for example, if I do a hand movement or a finger tapping, which is basically this function, every time my right hand works, there's a very specific region of the brain related to the coordination of this right hand, which turns out to be in the left hemisphere. Every time I do the same movement with the left hand, I have a very specific region of the brain on the right hemisphere. It's always the opposite when it comes to brain, right? But there are very specific regions that control any function that we have, be it a very simple, like motor task. It can be more complex that are very innate to humans, right? Like verbal fluence. Every time we keep talking about a specific thing, there is one specific region that is related to the language production, which turns out to be really on the left hemisphere, and that's for every single human, right? You can actually monitor different tasks, from physical exercise, if you like that, or reading, which is also a superior task, completely related to humans, right? So basically, be it MRI, be it with magnetic properties, be it with optical properties, you have a very specific way and a very good way to actually monitor brain function. And you do that looking at hemodynamics. That's basically the idea. Blood comes in and out, and hemoglobin going up and down all the time in the regions that we're looking for, right? So basically, my home message is actually, this works, right? So near-infrared spectroscopy, which is the optical spectroscopy, is a versatile technique that can probe the brain under different conditions in a wide variety of populations. People have been using that all the time, worldwide. So a lot of people are very interested in understanding which parts of the brain are related to which functions. And because if you know that, you can do a reverse order, right? So if I know that one very specific region of my brain is related to the movement of my leg, for example, and for whatever reason, I don't have a leg working anymore, but the brain is intact, so I can use the signal from that specific region to try to, for an exoskeleton, for example, and make a movement, right? That became very famous here in 2014 during the World Cup, right? So that was part of an opening ceremony. So basically, that's how you actually monitor. What you are actually monitoring is hemoglobin concentrations, which is an indirect way to monitor that, right? So that's what everyone does. But what I really want to talk here is a different thing, right? So we know that the brain changes every time we perform a single task. But really, I told you in the beginning that when we are doing nothing, right, it still consumes a lot of energy, right? So really, how does the brain work at rest when it's not performing any very specific and conscious task, right? So we know that something is going on because, well, some people have shown that if you look at the activity of the neurons and the other cells, glial cells in the brain, you have a lot of ATP usage, right? So there is a lot of energy even at rest, right? You have an idea, so every time you have a very specific function, it demands some energy. If you increase the amount of function, you increase the amount of energy needed. But if you go to the function to zero, you still don't go to the energy demand to zero. So even at rest, there is energy consumption in the brain to do something that we really don't understand, right? And that was actually the question that we like to try to understand, right? What's going on at rest, not when it's working during function, right? And the way we actually set up these experiments, it's a quite general idea, right? So basically, you have something that can collect the data from the brain, can be EEG, MRI, PET, can be optical spectroscopy. We do optical spectroscopy quite all of the time, right? So you can process this signal, you can remove artifacts from the signal. There are some specific things that you can do, and then you end up with a neural-based signal, right? So basically, be it if you're using EEG, you're measuring electric potential. If you're using optical spectroscopy, you are looking at hemoglobin concentrations. So you have a signal that's related to brain function. However, you're not doing anything, so you just have spontaneous fluctuations from these hemoglobin concentrations, right? You can do that at different regions of the brain. So here is just a schematics of the brain, and you have some dots that represent the fibers that we actually look, shine light and detect light. If you go to one specific region, what you see is just a time course of spontaneous fluctuations from hemoglobin concentrations. It's spontaneous because you're not inducing any activity. It's just what you're measuring when the person is just doing nothing, right? Then you can look at different regions, and you can compare the spontaneous fluctuations of this region to one given region that you chose randomly, right? And you can assign a similarity number, right? So you can compare these fluctuations with these fluctuations, right? So you can do that. There are a lot of different numbers. Some of them are more linear. Some of them are more non-linear. The most simple that we can think of could be the Pearson correlation coefficient, right? So that's one metric that you can measure. You can compare the two different time series, right? So you can do that for these two regions. You have a number that tells you how similar this series is with this series. If you look at a different region, you have a different time series, then you can still compare with this guy, and you have different numbers. If it's not related at all with the first one, you have a completely different number, right? A low number For example, if it's Pearson correlation coefficient, it has to be something between -1 and 1, right? So that's basically the same idea. So you can actually do this comparison at every different regions that you have in the brain, and you end up with a correlation matrix, right? Or a similarity matrix. And basically, if I pick one region, what I'm seeing is just one row or one column. It's symmetric. So one row or one column of the matrix. And if I pick this region and look at how the other regions are related, how the signal of the other regions are related to this region, I can actually go back and see how these spontaneous fluctuations are actually correlated or synchronized at different regions of the brain, right? So this is actually a functional map of similarity between different regions of the brain when the brain is supposedly at rest, right? So if you do that, and if you pick different places as your seeds to compare all the other regions, right, you end up, amazingly, with very specific, we call it networks or functional networks, right? So independent of the person you're looking at, independent of the technique you're looking at, what you end up seeing is if you pick a seed in the sensory motor region, which is kind of the middle up part of the brain, you see that there are very specific regions. They're always correlated with that region. No matter which person you have, which person you measure, no matter what you use to measure, it's always the same type of connection, the same type of network, right? And if you pick a different seed, you see a different regions of the brain that are always synchronized in these spontaneous fluctuations, right? So that tells you that there is a degree of organization in the brain, even when it rests, and probably this is related to the amount of energy. It does cost energy to keep these different regions of the brain synchronized all the time, even when you are doing supposedly nothing, right? So you actually can see that, and the idea behind it is there is some connections, and we know if you open the brain, if you go back to 1960s, 1950s, and open the brain, you see that there are different regions of the brain that are actually physically connected. The neurons from one part are connected to the neurons from the other part, right? But that's not what I'm talking about. It's a different type of connection that it's related to the function of the brain. And they do not follow the same connections that the structure has, right? So somehow there is a connection between different regions. They're not actually physically speaking. So there is information from different sides, right? From different parts of the brain that actually can be passed from one region to the other, which is not physically being passed, right? It's not really an anatomical structure, right? And you can actually measure that. So really, different regions of the brain are connected at rest, both anatomically and functionally, and that must demand energy, right? So the question that comes next, naturally, right, is how can you extract information from these brain connections, right? So you know that there are a lot of connections. A lot of people can measure that with a lot of different ways. And you always see the same thing. If you have someone that has a disease in the brain, you don't see these connections. So really, they are real. And so the question is, how can you actually quantify that? How can you use, how can you understand that? Because there is a problem, right? So if you get the same person and you try to do that different periods of time, different days, for example, for 3 different days, for example, you see that there is some fluctuation, right? So the map you get on the first day is not the same map that you get with the same person if you go the day after there and measure. So there is a fluctuation. On average, you see really nice maps. But if you look at kind of very specific time periods, you see you see variations of this map. You see fluctuations of this map, right? And that's what people really, a lot of people don't understand, right? So the point is, brain fluctuation is complex, right? And I probably don't need to go in detail with this slide, with this audience, but basically the idea is it's not when I say that the brain function is complex, I'm not saying that it's complicated, right? A plane is complicated, right? In order for a plane to fly, you need a lot of different parts of the plane to work, but then when the time that you actually have all these parts working, you have the plane flying, right? It's just the end result of this. When I mean, when it's complex, it's a different way, right? So I like these birds, and because it's basically, you have different parts of this collection of birds, right? Because you have different parts, each bird is a part, but basically, when you actually have all of them flying at the same time close to each other, what you see is a pattern that it's just not this individual pattern compared to other. That's the plane working, that's not the birds flying, right? And the brain is complex in this sense that basically you have different parts, they are all oscillating, right? And for some reason they are in phase with different regions at some of the time. So really, in order to understand that, what you need is to somehow take into account the interaction, right? So the interaction between the different regions of the brain. Right? And the way we actually do it, it's also not that complicated, right? So basically, you have the different regions of the brain that you can measure. From all of them, you can actually measure, for example, the hemoglobin fluctuations at rest. You can do that at different regions. You can have a similarity matrix compared to that. And then what I can actually do is define a matrix. It's a threshold. And I say, well, every region that is related above a certain threshold, I would say that it's connected, right? Every region below this threshold, I would say that it's not connected, right? So basically what you end up is with a graph, right? And I will avoid the term network here because in neuroscience, people refer to all these different regions connected, right, as networks, but they are not really networks in the sense of complex networks. They are just functional networks, right? So I'm just calling this graphs, okay? So basically, you can actually, by setting a threshold and looking the higher similarities, the regions that are actually connected with a higher similarity parameter than the threshold, you have this, you end up with this graph. And if you have a graph, you can actually compute graph parameters, right? So you can think of the number of links that you have in your graph. You can think of the average number of links, the degree of the graph. You can think of how the information actually goes from one side to the other, right? So you can actually quantify how all this information flows from one region to different regions in this graph compared by having the links between the different nodes, right? And since the threshold is arbitrary, right? I picked one arbitrarily speaking. What you can actually do is there's no simple reason why a single threshold is defined. So you can actually you can look at not only one threshold, but you can look at different thresholds like your order parameter of the system that you have, right? So that's basically the same idea. That's basically the idea. And what you can do, you can actually look at these different regions. You can see that when different regions, you can look at basically the region, the nodes on this graph are the regions of the brain. The links are how similar they oscillate in time. You can compare the density, right, of the links of the network. You can compare measures of integration of these metrics, right? So, for example, how far in terms of links one region is from another region, right? You can think of segregation of these graphs, right? So, are there clusters in different regions of the brain, and how they are actually clusterized, right? And basically, by characterizing different regions, right, or different information from integration, segregation of the brain, you can have different types of graphs, right? You can have, for example, you can go from very random graphs to very regular graphs, right? So they have different properties in these parameters, in the graph parameters, right? So if you have a random graph, you typically have low clustering in different regions. You have short path lengths from one region to another region, right? If you have a very regular, very well-behaved graph, what you see is a long path length from one region to a different region and high clustering, right? And in the middle, you have very specific types of graphs. You can have very specific types of graphs, right? So when you do that, very interesting to the graphs that you see in the brain, what you see is actually these properties are remarkably similar for different people, right? So this is an average of about 100 people that we measured over time, right? So you see basically that if you look at the networks that they have, so I'll just go back, right? So just to remind you what we are doing, we are just measuring different regions of the brain. They are the nodes of our graph. We are comparing the fluctuations in their hemodynamics, and the similarity between two different regions are the lengths. And we are doing that varying the threshold, right? So basically, you're not looking at just one graph, but you're looking at a lot of graphs depending on the threshold you have. Okay, so for every single threshold, if you have one graph, you can compute the parameters, and that's one point in this graph, right? So then you vary the threshold, and you have a different graph that will have different number of lengths, different correlation coefficient, different degrees, different path lengths, different clusterings, right? And if you do that and compare these parameters for different people, what you see is actually, independent of what is doing nothing for these 100 people, their graphs have very similar parameters, globally speaking, right? Comparing all the different regions of the brain. Right? You have a variability across different people that is less than 20%, right? However, if you look at the same person, one person, at different times, what you see is actually they have very different connectings. They have very different connections, they have different graphs, right? It's the same person measured 4 different times. And you see 4 different graphs. But all these 4 graphs have the same global properties there, right? So put it on a statistical mechanics language, maybe. Basically, you have different microstates that the brain can stay, right? But they are all they all yield the same macro state, right? Which can be measured by these parameters, right, that you have. So, and that's another indication why, or another reason of why possibly you need so much energy when you're doing nothing, right? Because you can actually fluctuate from different microstates from one time to the other. They all cost the same in terms of connections, right? But you're always fluctuating from different times of the other. And if you think really deeply about that, it does make sense, right? Because when you are doing nothing, you are about to do something in a moment later, and you can do anything, right? So you can be actually prepared to do anything if you are always rotating in different microstates, right? So that's basically one way to see it or to interpret that, right? So basically, another message is actually, despite the local variability in these spontaneous hemodynamic fluctuations, right, what we see is actually strong intra and intersubject similarities of these global properties of the brain, right? So there are actually very similarities. There are fluctuations from the same brain, but they all lead to the same macro state, and they appear to be the same macro state independent of which subject you are talking about, which person you are talking about. Right? So we are, biologically speaking, very similar in terms of how our brain works and how it costs, right? Which is, I mean, it's not surprising, right? In the sense, since we are all from the same species, right? What's nice is actually you can measure that and you can quantify that, non-invasively speaking, right? And then the question comes, well, where does this behavior come from? So I already made a kind of similarity or analogy to statistical mechanics, so I can go even further, right? I can think of the simplest system that we have that we know that can interact between different regions, right? So you can think of, for example, a simple system of two states, right? And you have different regions. You can think of different spatial regions here. And you can think basically, well, if you have two states, you have an energy that it costs to be in one state or the other state. And you know that globally, any system would actually minimize this energy. So you can just randomly assemble one random state to every different region. And you can compute the energy of this system. And then you can let the system evolve in time. That's basically the same idea of an Ising model. If I just think of these two states as a spin up and spin down, right? So I don't need to think of a system with spin ups and spin down. I can just think of a two-state system and assign an energy to any of these two states, right? So that's basically the same idea that you do in the Ising model. And you can evolve over time. So what you can do is actually the same procedure that we've been doing with neuroimaging data in the brain, right? So different regions, you have a time course of the state, either state one or state two, right? And you can look at that over time. You have a time series for every different region from which you can build a graph, right? But we know from the Ising model that there is one parameter that dictates these connections. In the Ising model is basically the temperature, right? So if you have high enough temperature, it's easy to go from one state to the other. If you have very low temperature, you basically stay in the lowest state possible, right? So this is basically, temperature is just basically the probability of switching from one state to the other. And that you can control with a very simple probability. So I can go back to this model and assign different probabilities, right, that I can switch from one state to the other. If the probability of switching from one state to the other is very low, so basically, if you look at one specific region here, over time, it's going to be just a very boring time course, right? There's no switching. If you have a very high probability of switching from one state to the other, then you have a lot of ups and downs, a lot of flipping, right, from one region to the other. And if you have a very specific probability of switching from one to the other, right, which is, I call it a critical probability, which is similar to the critical temperature in the Ising model, right? So what we end up seeing is actually, well, there are probabilities of flipping, but if you look at spatially speaking, you have patterns that it's just long-range patterns, for example, right? So you start forming really brain patterns, or not brain here, but well, spatial patterns, right, that you can compare. So you can go for different probabilities of switching. You can see a time series. You can build a graph on that, and you can compare this with the graphs that we have from neuroscience, right, from the hemoglobin oscillations. When you do that, right, and you compute the graph parameters, what you see is actually, if you have a very high probability or a very low probability, that will give you all of the time for any of the thresholds you are looking at, graph parameters that are very different from what you have when you measure the brain, right? But if you are at the critical probability of switching, right, you end up with graphs that the graph parameters are very similar to the graph parameters that we measure in neuroscience, right? And for every single threshold that you want to see, right? So, which tells you that really, if you want to try to understand what this graph is coming from, it's probably you can actually model that. I'm not saying that the brain works like that, but you can get very similar properties that you have from the brain from a very simple system, a very simple two-state system that flips at the critical temperature or the critical probability here, right? So that, again, offers another possibility of interpretation of how the brain works at rest, right? It's flipping from, it's always flipping at different connections, at different types of graphs. They all have the same global property, right? And you can actually get this if you're thinking of switching at the critical temperature, at a critical probability, right? So this is, I think it's very exciting, right? So basically, the behavior of the global network properties measured with optics, all the data that we collected is from optics. People have actually done that with fMRI as well. And basically, it can be produced by a system of interaction between closest regions at a critical transition probability, right? So okay, we know there's a right degree of switching, and it's a kind of critical degree of switching. And we are always switching, our brain is always switching different connections all the time at a very critical state. And this yield very stable global properties of the brain, right? But the question is, well, if they are so stable, the next question is, how do we learn, right? Certainly, if you actually measure that we measure that for 100 adults, right? And more than that, we are in a university, so 100 university students, right? So, but if we get different phases, right, we typically learn, and we always associate that with different connections in the brain. So basically, if we learn something, we should actually change the model of something, right? So the idea is, can we actually see that, right? Probably in an adult, your brain connections are formed already. So to answer this question, we actually went to babies, right? So basically the idea is, if you look at a baby, right, right after they are born, you can measure their brain connections at rest as, well, the best that a baby can be at rest, which sometimes can be crying a lot, right? But if you measure that, that's the time period of our life that we are always learning new things at a time, right? So we did that with babies. We measured them at 6 months old, and then we measured that at 1 year old, right? The same baby. Okay? And then we look at the properties or the graphs that we obtain with every baby at 6 months and every baby at 12 months. And this is just sample random graphs that we get from one of the babies. But the point is, if you actually have the graphs from one baby, well, first of all, the pattern is still the same. All babies at 6 months old, they have very similar graph properties, right? And all babies at 12 months old, they all have the same similar properties. But when you compare the properties from the 6 months to the properties at 12 months, then you start seeing changes, right? And they are mostly, these are global changes, but you can actually look at that at very specific regions of the brain. And the major region that actually you see changes are around the motor cortex, right? Which is related to, well, movement, right? And that's typically what a baby from 6 months to 1 month, to 1 year is concerned, right? It's the primary concern of a baby. That's about the time they start walking and well, trying to live by themselves, right? So basically, yeah, it is stable, but as we learn, we are actually increasing the number of connections. We are actually increasing the complexity of this information across different regions of the brain. And we can see that from babies, right? But then there is another philosophical question that we actually think of, right? So, okay, we measure a lot of people. We measure 100 people. They all look the same, or the brain patterns look the same. We can actually have a very small variation. But we know that everyone is unique, right? So we don't really think of, there must be something that characterizes every single human being, even when the brain, right? So the way we think, the way we act, the way we move, and things like that. If that is true, right, so it has to be very difficult to measure because one person keeps having different connections profiles, right, from every time to another time. The global properties are always the same from the same person. But the question is, are there links in these different ways of the brain to rearrange itself? They are very specific to every single person, right? So that's a very complicated tech question. And what we end up doing is, well, if there are links, we cannot see it. It's not very easy to see it. So we actually went to maybe a neural network can see that, right? So we end up having, well, we know that this is the similarity matrix. We measure 100 people. We measure them at least 4 or 5 different times. So some of the people we have 6 different times. So we know more or less the profile of 100 different people, right, from at least 5 to 6 different times. So the question is, if we have this information and we give a neural network this information, can it just classify or identify which subject I'm talking about? Right? So we use the data from all these 100 people, part of this data from 100 people, and then we give, oh, this is a set of brain connectivity at rest that we have from one person. Can you guess which one it is, right? And amazingly, if you do that, and if you get all the profiles from oxyhemoglobin and from deoxyhemoglobin, you can actually classify correctly about 97% of the time, right? So that tells you that, yes, there is a very specific pattern with a very global metric, right? For every single person, it fluctuates in time, but it does not fluctuate very similar, or it's not the same fluctuation for every single person. There are very specific links that we can actually assign to every single person and correctly classify that about almost 100% of the time. It's like a brain ID, right? So everyone has a brain ID from the different set of links across these 4, 5, 6 different periods of time, right? So yeah, despite similarities in the spontaneous activity fluctuations, the networks that we see, the graphs that we see, are intrinsically different, right? So, but there is still an open question that we still don't know the answer, right? So basically, right? So we know this can change, and we saw that for babies. But the question is, we know that most of the times the brain can be forced to change. And that's a very well-known phenomenon in neuroscience called brain plasticity, right? Something that is plastic. You can model differently if you need to, right? And we know this is a phenomenon that occurs with different people. For example, if you have a stroke, right, and it affects the ability to walk, right? Somehow the brain knows, or it finds a different way to learn how to walk again. It might not be that efficient as you had, as the walking that you had before the stroke, but we learn how to do that with a different region of the brain, because the region of the brain that's related to the walking is that after the stroke, right? So there is a phenomenon of plasticity that the brain can arrange itself. And the question is, can we actually see that? Can we actually measure that? And above all, can we actually predict it? Because if we can predict it, we can control the process, right? And then we are talking about a whole different way of physiotherapy. So PT, physiotherapy, the way it exists now, right, wouldn't be needed if I can control the system based on the brain connections, right? So this is still an open question. We don't know the answer. We are actually measuring people with stroke right now, currently, right? So, and try to see this pattern over time. So there's still a long road ahead, and we don't know the answer yet, but in the end, it may be worth the journey, right? So with that, I'll just finish and acknowledge my group, all the data that I showed you, and it's a lot of work, mainly to collect all this data, is done by my students, postdocs, who are actually working with me. And all of this is also collected with different regions and different institutions, right? So these are all people that are somehow related to some of the work that I presented to you. And more specifically, I just want to acknowledge my colleagues and students are with me for over 10 years now at Unicamp. All the baby data was collected in Belo Horizonte at the Federal University of Minas Gerais under Deborah and Marco. And all the data that we have with stroke, and some of the data that we collect with the healthy subjects was collected in Philadelphia at the University of Pennsylvania with Arjun, Joe Dutcher, and John Greenberg. So basically, just this is the lab we have. We call it the Biomedical Optics Lab. If you want to know more information, we have 2 web pages. The web page in English is liveforbrain.com. The web page in Portuguese is luisacerebro.com. And we also have a Facebook web page. It's called LOB, from Biomedical Optics Lab in Portuguese, Unicamp. And this is basically some of the things that we do. It's not all of the things that we do. So basically, this is related to the functional connectivity of the brain, how the brain is connected. We try to use that and translate that to clinical applications, including photodynamic therapy, which is a therapy to treat cancer. We also have been trying to use that to predict connectivity in function. And with that, we can design brain-computer interfaces that can control, you can control basically any interface just with your brain data, right? So that's basically the main idea. And even modulating your own brain based on these connections that we see, right? So I guess right on time, I'll just finish. I want to thank you for your attention. If I have time for questions, I'll take it. Thank you.
So thank you, Professor Rickson. We have time for questions. Actually, I have one question. You showed image of the brain when you speak, some areas of the brain. When you speak different languages, are these areas the same or do they change?
They do change, but they do not change dramatically. So if you go back there, something that we actually don't appreciate much, right, is how complex reading is, right, or speaking is. So because you have a lot of different regions that work simultaneously for kind of the speech the way we know it, right? So there are different, some of these regions are just biological regions related to speak, and whatever language you speak, it's always going to be the same. But one small part of the brain, more anterior here, they do change, right? So this is related more to as a kind of first language and other languages.
And for example, if I move my hand, there is a region that activates. And if I think about moving my hand, is it the same region that activates?
That's a debate. It is the same region. However, it's a weaker signal, right? So, yeah, if just thinking about doing it, we will also activate the region, but it's going to be a very mild activation. So to the point that it's very close, it's very hard to detect. But if you do it, and it's very hard, not because of the brain, because the ability that we have to measure the brain, right? It comes out with a lot of noise. So you need a lot of signal processing to look at that. But yes, it does activate the same region.
Okay. And your conclusion is that the brain works the same in microstates, independent of the person?
Well, that's what we see. For 100%, a lot of them Brazilians, a lot of them Colombians, a lot of them Americans.
And different sexes. Because yesterday we had a discussion that perhaps the brain is different, works different.
Yeah.
So,
Well, unfortunately, I cannot answer this question with my data because, well, I'm in a physics department which is heavily heavily weighted towards male. Right?
I saw your group picture.
Yeah, right. So we have most of our data. It's not a kind of 50-50. It's heavily weighted towards males. Some people who actually look at these gender specifically differences, they do find different patterns in the micro states, but the macro state is the same too, right? So most of this difference from this 18% difference that we saw across different subjects, they do not come from gender differences, right? So, yeah.
Very interesting. Agora a gente sabe, ele não. Any questions for the audience?
Yes, there's.
One question related to your last statement. You said that there is some kind of difference in the macro state if you test for different sexes. But is this kind of measurements performed for young people or, I mean, because then you go to some social bias, I would say, and could develop different.
Yeah. So, well, social in the sense of the age-related differences. Yeah. So there are age-related differences. Here we're just showing data from young people, right? So it's mostly between 20 and 30s. If you looked at the same profile with 50s, 60s, you see differences. And interesting, you see difference in the macro state as well. So there's a decrease in the connectivity in the brain and a decrease in the path length of the graphs, right? And another interesting question that people have been asking is maybe we can use that to see aspects of aging. That's true, but also we can use that to treat or to diagnose at least or to anticipate neurodegenerative diseases like Alzheimer's. Alzheimer's is probably the most famous, right? So, yeah, so there are differences, we know. If you look at 20, 30 people, you see one type of macro state that yields different parameters. If you look at 50, 60s, you see a different macro state, mostly with lower information flow, lower connection density, right? So, yeah, we see. And the other interesting question that we can answer is, even in the adult life, we are learning, right? People can learn, right? So the point is, all these different photos that we see from the brain connections, they are all from 1 week apart, right? So it's not very different term in kind of in a timeline. So the question is, if we actually look at a 2-year time frame, so does that actually change, right? So we are trying to do that now, but it's incredibly hard to have people to commit to get us the brain scanning, right? Every week for 2 or 3 years, right? So, yeah, but that's a question.
When you compare the IC model with your data, the critical exponents also match precisely with the critical exponents of the IC model?
Yeah, so because it's the same state, we had to use the same critical exponent, right?
Right. So it's in this universality class, right? You know that this fixed point describes lots of things, not only IC model. So what is the other parameter?
What's the parameter?
The other parameter, the up and down. I don't know.
No, so yeah, so that's the point. It's the way we used, or the way we actually look at the IC model here is it's like a toy model, right? So we can't really explain what the parameters mean in the brain, right? So basically, we have a model. It's a two-state model, right? And basically, these two states is related to that region is active or not active.
Okay.
Right? So hemodynamic speaking, right? Active and not active because we are assigning oxyhemoglobin going up or oxyhemoglobin going down, right? And we know that every time oxyhemoglobin goes up, it has a kind of brain region active in the sense that the neurons in that region are spiking. What it really means, even the parameters that you collect from that. So for example, in the Ising model, right? So we do all the math and then we calculate the magnetization, right, of the sample. So we don't know what, we could calculate that Chuhi, right? So, but we don't know what it means.
Okay.
Right.
But the correlation length, you know what it means.
The correlation length, we know what it means.
And it matches so with that, I think you can extract the critical exponents already.
Yeah.
And
so these numbers, they do match.
They do match. Okay, so
match.
this was my question.
Yeah.
So it doesn't need for Saturday class.
Yeah, exactly.
Okay.
All right. Thank you for the lecture. When you select the people, also you take a look on people that has different cognitive recognition, like for example, dyslexia. We know that people with dyslexia, they have a different way of thinking and other things. And when you select the person that you did the experiment, you identify people with this kind of
No, we actually exclude them before we collect the data because for every one we call it a healthy subject. In order to understand it, what a healthy subject means for us is basically someone with no neurological diseases that we can collect from the past historical data. But they all go through an interview before we collect the data. So we can pick up, for example, dyslexia. We can pick up different aspect, coordination or lack of coordination. We can pick up these kinds of things. Aphasia in speech. So, and we typically discard these subjects. All right.
Hi, I think it's a more basic question, fast question. Does this right hand, left hand graph tell something about the person being right-handed or left-handed?
It does. It does. So, first of all, what you're seeing in the screen, no, right? So they are all what we typically do is get the hand dominant to do the experiment, right? So if you are right-handed, you are doing a right-hand experiment, and if you're left-handed, you are doing a left-handed experiment. So that way we can control for the dominant hand. But you are right in the sense that if you are right-handed, your profile of activation in the left hand is going to be a little bit different from the profile that when you activate the right-handed. And the major difference is actually when you are right-handed, you are doing a right task. You have a very contralateral response in the sense that it's very focused on the other side of the brain, right? If you are a right-handed and you are doing a left-handed task, it's more bilateral. So you see a response on the other side of the brain, right? And that is explained by the structure of the brain. But you also see some activation in the same side of the brain, right? And that cannot be explained by the structure. So that's why.
But based on what you said, couldn't you tell that this person is left-handed? Or
no. So basically, this is a group result of over 200 people, right? So basically, yeah, and they are all right-handed or left-handed. Right. So that's it's just an average response. So the color scale is just the frequency of activation across these 200 people. But if I had one person, I could actually say. It could be easy to say that.
Thank you very much for the talk. So when you compare the networks in the Ising model, when you did this, did you consider the functional network, the connection, the structures that you said, the length path and the connections? Did you consider between regions, or because you can separate between different regions of the brain, but each region also has its own graph? I'm not sure if I could explain.
Yeah, I'm not sure I understood it, but
because you have different regions in the brain, and these regions are connected, but the neurons in these regions have also different properties. So connections and
they have different. So what you're saying basically, there are two different ways that you can build this network, right? So one way is to look at the functional regions, and that's what we've done, right? And that's what it matches actually the Ising model. There's another way to do it is just discarding all the functional information and building the connection of the physical connection of the neurons, right? So people call that the structural connectivity maps. We can't, well, so here's the thing, that's not what we have done, and therefore I cannot answer whether that would actually match a functional. I would suspect not, because they're completely different, but we haven't investigated that. One of the main problems of trying to do that is it's very easy to track these connections, these physical connections, in very small animals, right? Yeah. It's very hard to track these connections in humans. So the best approach that you can do that is with an MRI technique called tractography, when you actually look at the nerves, and there are it's basically a connection of the population of the axons of the neurons. But there is a lot of controversy on the accuracy of this data. So I don't think anyone has actually investigated that deeply or accurately to answer that.
And do you plan on studying people with some neural disorder?
Yeah, we are very interested in trying to do that. But at the same time, it's very hard. So we have to define. We talk to a lot of physicians, right? So my background is physics, so I'm learning all this new field of medicine. And there are different questions that even neurologists don't know what they want to look for, mainly in this, for example, Alzheimer's, right? Because one question is, yeah, you can characterize Alzheimer's, and you can see most likely a very dramatic change in the number of these macro parameters, right, of these macro states. But the real important question for the clinician is, can you predict it, right? So, but in order to predict that, you have to do an experiment that you know that in a few months or a few years from now, that person will actually have Alzheimer's. And they don't have this information, so it's very hard to actually enroll people to collect the data, and it's very inefficient. So that's why what we prefer to do is we go and we collect data from 600, from 6-year-old people, and we kind of track them down over time, but it's a 10-year work. So yeah, but we are very interested in trying to understand that.
Thank you for the representation. It's possible to make any predictions about the future behavior of a person just looking for her or his brain?
Oh wow, I don't know. We haven't tried that, but I would bet it would be really hard, right? So it's kind of like Minority Report, right? So like Tom Cruise's Minority Report. I don't think it would be possible. Well, it might be, but I wouldn't bet on that based on the brain, because I mean, we know we can change, right, the time course of what we do, right? If I have a stroke now, I know that my brain will have to change completely, right? But I don't think you can predict that. And even kind of a very specific strokes, right? Because we've been measuring them. And it's not very simple in the sense that when someone have the same type of stroke at the same region that affects the same function, right, like speech, the way that the brain reorganizes appears to be completely different for every single person, right? So so based on that, I would say that it's very hard. It would be very hard to actually predict the behavior on these brain connections.
All right, so let's thank Professor Rickson again.
Thank you.
If you have more questions, perhaps you can talk to him during the break. There are food and some refreshments upstairs. And we have half an hour, so we are 15 minutes late.